I'm Merlus. I'm from Tuskegee University, double major chemistry and chemical engineering, and today I would like to present my research conducted at the University of Minnesota during the summer of 2017. Today, here's the objective that I just want to cover with you guys. First, I want to address the importance of addressing my research question, give you a little background, discuss the methodologies, and conclude with the conclusion and results. The importance of addressing the research question. Today we commonly use quantum dots, and quantum dots are used primarily for various applications, as she mentioned earlier. And we know the bulk material for most quantum dots is cadmium, lead, and other toxic material. So we want to go ahead and try to use the same, we want to go ahead and try to benefit from the same applications that we find from quantum dots, however, using our doped silicon nanocrystals. And here's a few of the disadvantages I want to address, which is the very toxic, lower quantum yield compared to the doped silicon nanocrystals. And our doped silicon nanocrystals are low cost and earth abundant and size tunable luminescence, easily synthesized via non-thermal plasma reactor. And if you take a look at this image here, this image illustrates the research question that we're trying to address. We theorize at some point that our doped silicon nanocrystals, in whichever application that they're used for, will be found in the landfill as propped by this R&B singer named SZA. So notice that these are just computers, and you know that computers have semiconducting material. And the semiconducting materials somehow interact with our environment, where we find naturally abundant bacteria such as Shigella odensis and other organisms that we find in our environment that are beneficial. So we wanted to go ahead and explore what would possibly happen if our material were to interact with this organism. And our model organism of choice was Shigella odensis MR12. To give you a little background, what are luminescent doped silicon nanocrystals? You know, silicon nanocrystals come from the bulk counter material silicon, and silicon nanocrystals are material that absorb light and re-emit light based on whichever size or whichever wavelength that it is absorbing. So applications of these doped silicon nanocrystals vary from different applications. However, I'll go ahead and discuss that in further slides. Here's the plasma reactor used to go ahead and synthesize these doped silicon nanocrystals. I myself was not responsible for synthesizing these doped silicon nanocrystals. This project was a collaborated project with the mechanical engineering department at University of Minnesota and the chemistry department at University of Minnesota. And this wonderful process is crowned by the scientist that goes by the name of Dr. Uwe Kortagin. Applications of the doped silicon nanocrystals. They vary from biosensing, bioimaging, photovoltaic, photothermal therapy, and as you see this image here, you have the RDFI signaling used for, biosensing. You have your photothermal therapy. What is photothermal therapy? The photothermal therapy is using electric radiation to go ahead and focus on a particular site, a tumor site, to go ahead and eradicate that patient or whoever that is undergoing or that is experiencing this illness. And as I mentioned, there are various applications that you could use with these doped silicon nanocrystals, from optical, electronic, nanoelectronic, photoelectronic, photovoltaic, and sub-wavelength microscopy. Okay, silicon nanocrystals and doped silicon nanocrystals, they're different. So the doped silicon nanocrystals, how they differ is we could go ahead and use quantum confinement in the silicon nanocrystals, as Andre mentioned earlier, to go ahead and change that luminescence that we observe. So if we change the size of the particle, we could go ahead and observe different color waves. However, when we dope it via N-type or P-type, we could keep the particle size and still observe those different wavelengths. So my role this summer was to go ahead and evaluate whether this material that was being synthesized in the mechanical engineering department was toxic, and our model organism of choice was Shunella odensis MR1, MR magnesium reducing MR12, actually, excuse me for the 12 missing here. And that organism is found in the soil sediments and originally found in Lake O'Donnell in New York. And this organism is the model organism of choice for most nanotoxicity studies, such as the lead nanoparticles, nickel nanoparticles, and any other nanoparticles that are being evaluated for their toxicity. And this study that I'm getting ready to display with you guys here is we observed the dopant via P-type, which is just doping it with boron. So research question being addressed: how will the P-type boron dope silicon nanocrystals in varied concentrations interact with Shunella odensis? Or how would the N-type phosphorus dope silicon nanocrystals in varied concentrations interact? Why do we want to vary their concentrations? Because when we vary their concentrations, we change the luminescent property. Okay, so we don't have to actually change the size of the particle, but we increase the concentration of the dopant, and then we go ahead and observe this photoluminescence property. So the method used was the drop plate. This is just a preliminary test being conducted. So if we determine that our particles are not, our dope silicon nanocrystals via P-type is non-toxic, we can go ahead and conclude that we don't need to do any further analysis. However, if we do conclude that they're toxic, whether it's P-type or N-type, we will continue to redesign our dope silicon nanocrystals until we observe a non-toxic. So the goal was to explore the potential effects of the dope silicon nanocrystals on our model organism. So this characterization TEM technique was provided to me by the courtesy of my graduate mentor at the University of Minnesota. And as you notice here, in this low resolution image, there's an agglomeration here that happened because of the dope silicon nanocrystals were placed into a solution of water, and the lack of solubility of our dope silicon nanocrystals caused this agglomeration here, aggregation here, sorry. And this high resolution image here shows a better picture of the aggregation of the intrinsic silicon nanocrystals, which are the undoped silicon nanocrystals. The XPS image here is just, the XPS graph here illustrates basically the chemical composition of our intrinsic silicon nanocrystals, our undoped silicon nanocrystals. And we notice here there was a carbon peak that appeared here, and that was a result of the tape that was placed on the filtration to receive the dope silicon nanocrystals from the plasma reactor. So this is accurate. This is about what we were looking for. The technique we used was the X-ray photon survey. Methodology. Going back, as I mentioned before, the drop plate assay was the method that would be used to go ahead and determine the toxicity or explore the toxicity of these dope silicon nanocrystals. So first I had to prepare my solution, grow my bacteria to optimal density of which was about 0.5 and 0.5 milligrams per milliliter. And from there, we were able to section off our different concentrations. Well, this is the negative control. In our negative control area, we will have just about the highest concentration of our bacteria. However, once we go to the fourth region and fifth region, it would be a low concentration, so we will be able to quantify and to be able to predict how much bacteria we should have in each section, right? So basic method: prepare our bacteria, centrifuge it, DPS, use our buffers, and from there we have our bacteria. Once we have our bacteria, we prepare our working solution. Our working solution was just basically water and our dope silicon nanocrystals, as I mentioned, from our collaborators in a mechanical engineering department. Here's another characterization technique provided from my graduate mentor, Bo Z. And it shows a dark field image of what actually is taking as the aggregation due to lack of solubility. So the brighter peaks are just basically showing us, okay, as we noticed when it's in solution, parts of the silicon nanocrystal was harder to dissolve in the water opposed to other parts of it. And that actually caused it to be a little more difficult to pipette and to transfer to the 96 plate well that you'll see now. And here's just a low resolution image of what's actually taking place. Again, here's just a reference of what we should be looking for once our doped silicon nanocrystals are exposed to our model organism. And notice that our model organisms are just some small rods, and you can see that this is just a control. And notice that here, when it was exposed to our doped silicon intrinsic silicon nanocrystals, that you don't see that many rods, you don't see that much bacteria everywhere, because this was a result of the lack of density that I failed to ensure that my density was optimal. And this is not a result of the toxicity of the doped silicon nanocrystals. I don't want you guys to take that from the image, but this is just the result of me not having the optimal density to observe what was actually going to take place.
So is that from photo microscopy or what?
This method here was actually the hyperspectral imaging. Okay. And again, this is the method used to go ahead and prepare the drop plate assay. We had conducted a tenfold series dilution, and this is what I meant by, once we have the fourth region, it will be less. The third region will be the amount of bacteria that we expect, and the fifth region would be another a quantifiable amount. However, if we would go ahead and try to experiment on this region here, it would proliferate a little too much, because the bacteria here is like at the maximum amount of bacteria that we grew. We don't want that. So here's a quick image of what actually took place once the drop plate assay was conducted. Well, we conducted two different time exposures, 30-minute and the 2-hour exposure. During the 30-minute exposure, it appears that the proliferation happened quite well, and notice that in the fifth region, it's like less bacteria. That's what we wanted. We expected it to have less bacteria. We expected to have a quantifiable number, like we able to count each colony. And notice when we get to the more concentrated area, it's harder to count. Again, this data is a bit inconclusive due to the fact that it was only a 10-week summer, and I had to work with collaborators across the lab where the plasma reactor was not easily accessible, so I only had like about 3 batches of doped silicon nanocrystals. Had to treat it like gold, failed doing that. But moving forward, here's the 2-hour exposure that took place, and again, results were inconclusive due to some human error, or me neglecting to ensure the optimal density, or maybe it is toxic, maybe it's not toxic. Don't take that away from this, because like I said, this was only conducted during 10 weeks, and this is what I actually was able to observe. Here's some more images of, here are the intrinsic, the doped silicon nanocrystals at 5% boron. And notice that it's some proliferation happening except here. 2-hour for replica plate 1, but replica plate 2 had some proliferation. So it had to be some type of error, because they're not matching. They were conducted at the same time. So in conclusion, doped silicon nanocrystals were synthesized via non-dermal plasma reactor, characterized using these various techniques, used desired cell density for the drop plate assay. We were able to count colonies. However, there must be at least 2 experiments conducted before we could quantitatively analyze the nanotoxicity of these doped silicon nanocrystals on our model organism. Future work, we will ensure that our doped silicon nanocrystals are being exposed to the optimal cell density, which is about 0.05 times 10 to the 7 cells per milliliter, and we will go ahead and assess the toxicity of the various concentrations. Notice that the previous one I just had 5%, so we want to go up until the concentration of 30%, and because at those percentages the photoluminescence also changes. And we're going to also conduct the same characterization techniques to go ahead and determine whether or not we have the material that we need. Again, I want to acknowledge the wonderful people at University of Minnesota for assisting me in this process, and I want